The first feature of the adenylate cyclase system is the receptor that initiates the activation of the enzyme adenylate cyclase and the second messenger it eventually produces. One of the most important receptors that is associated with the activation of the adenylate cyclase system is the glucagon receptor. This receptor can only be activated by the hormone glucagon. The second important feature of this system is the enzyme itself, adenylate cyclase. Adenylate cyclase is responsible for the cyclization or the production of cyclic AMP from ATP. This molecule is considered a second messenger when it is produced inside the cell. The third important feature of the adenylate cyclase system is the identity of the G protein involved in the activation of the target enzyme adenylate cyclase. The identity of the G protein is designated Gs-alpha. The S designation for this G protein represents stimulatory, meaning that this G-alpha subunit stimulates the adenylate cyclase system. Upon activation of the glucagon receptor by glucagon, the activated receptor associates with the trimeric Gs-alpha-beta gamma subunits and leads to the dissociation of the beta gamma subunit from the Gs-alpha subunit, followed by the exchange of the GDP bound to Gs-alpha by GTP. Gs-alpha with GTP bound to it is now able to associate with the membrane-associated target enzyme adenylate cyclase and can stimulate its activity to produce cyclic AMP. The fourth important feature of the adenylate cyclase is the identity of the second messenger in this system. The identity of the second messenger in this system is cyclic AMP, which is produced by the action of adenylate cyclase on ATP. Cyclic AMP is considered a second intracellular messenger because its activity was increased due to receptor activation and its concentration as a result inside the cytoplasm rise, whereas its concentrations were very low prior or non-existence prior to receptor activation. The fifth important feature of the adenylate cyclase system is the cellular responses that are generated by cyclic AMP. Cyclic AMP can produce two types of cellular responses. The first type of cellular response is usually quite fast and it involves the activation of a protein kinase designated protein kinase A. Cyclic AMP binds to protein kinase A and activates it, and allows this protein kinase to phosphorylate specific enzymes or proteins, resulting in a cellular effect. Another slower effect of pKa involves its ability to activate a cyclic AMP responsive element binding protein, which is essentially a transcription factor that can activate the transcription of specific genes. If you like this video, please subscribe. Thank you.